So now that we've covered the cells, we can jump into the different layers of the epidermis, also known as stratums. So if you look at the picture on the right, you can see the basement membrane, or what we call the dermo-epidermal junction. This time I'll do it in green, where you can see the tissue just drastically changes. So this would be epidermis, and this would be dermis. So in the epidermis, we have 4 to 5 layers. It depends on what type of skin we're talking about. If we're talking about thick skin with the palms of the hands and the bottom of the feet, we have all 5 stratums present. And you can see here we have the order. We have the stratum corneum is the very, very top, or most superficial. That's actually what's touching the outside environment, if you will, or what the air is surrounding. Then we have the stratum lucidum. Followed by that, a little deeper, is the stratum granulosum, then the stratum spinosum, and the stratum basale, or that basal layer. Now, this is thick skin. Thick skin has all 5 layers. In thin skin, what happens is usually the stratum lucidum is not there, and all the other layers are just a little thinner. They're just not as thick. So with the thick skin, all the layers are a little thicker, and then we have this extra layer called the stratum lucidum. So, do you have to know the order of the epidermal layers? You do, yes. But it's real easy to remember. Just make a mnemonic, and you can figure out a way to put these in order, just like we did with PMAT, with mitosis a couple chapters ago, right? Prophase, metaphase, anaphase, telophase. All we have to do is, if we take the first letter of each layer, let's just make up some type of rhyme or some type of saying, and it'll help you remember it. So the one that I was always taught was, come, let's get some bacon. And if you remember, come, let's get some bacon. Yes, it's silly, but you know the order. It goes CLGSB, corneum, lucidum, granulosum, spinosum, basale. Now, once again, in the case of thin skin, though, this stratum lucidum is not present. So in that situation, we would just come get some bacon. So those are the 5 layers, and what we're going to do on the next 5 slides is talk about each specific layer and what's unique about it, why they're important. So let's work our way in order, going from superficial to deep. So come, let's get some bacon. Come, the first one is stratum corneum. And remember, those corneocytes at the very, very surface of the skin, they make these shingle-like appearances, and they kind of look like horns. And you can see couple of these horn-looking structures at the very top of this micrograph right here. That's where they get the name. So this is a layer. It's actually several layers. They can range up from 20 to 30 layers thick. This is by far the thickest layer of the epidermis, the corneum. And what these are all dead cells. These are all corneocytes. So all those keratinocytes, excuse me, pump the cells full of keratin, they killed them, and now basically what they're doing is they're just migrating up to the surface, getting ready to be flaked off. So all cells in the stratum corneum are dead. They do not have a nucleus, they don't have active organelles going on inside the cells. Now, this once again does provide a waterproof barrier, and also just a barrier in itself from debris, bacteria, fungus, all that stuff as well. And this is the stuff that you actually shed. When you scratch your skin or when you roll over in bed on your pillowcase and on your sheets, you shed this stuff. And the average person, believe it or not, sheds about 40 pounds of skin in a lifetime. So depending on, average age, you shed about 0.5 pound a year, maybe a little bit more than that, of dead skin cells, of these stratum corneum cells in a year. So when you're, and also when you're looking at somebody, when you're looking at your wife or your husband or your boyfriend, girlfriend, whatever, you're mainly looking at a bunch of dead cells on the surface. That's what it is. So they have the saying, beauty is only skin deep. It's kind of funny because most of the cells that you're looking at, and if you're touching somebody's hand, they're all dead. They're all dead cells. And that's the stratum corneum. Next, we have the stratum lucidum. And the stratum lucidum, very similar to the stratum corneum, is just a bunch of dead cells. Just a bunch of dead cells. But remember, the unique characteristic of the stratum lucidum is this is only found in thick skin. So it's only found in the palms of your hands, soles of your feet, some fingertips, right? That's it. Or when I say some, all fingertips. And the reason why we get this name stratum lucidum, lucidum actually translates into light. So it's this lighter, more transparent, clear layer found just under the corneum. And its main job, yes, even though the cells are dead, it's just extra layers, extra layering for, once again, protection. Moving right along, we have the come, let's get. So G is for stratum granulosum. And the reason why it's called granulosum is because we have a bunch of granules, these little like specks all throughout the layer. And that is part of the process of making a whole bunch of keratin in this layer. Because, my friends, this is where keratinization occurs. This is where we are actively starting to pump the cells full of keratin for protective purposes. But remember, the side effect to that is we're killing the cells, essentially. So the two layers above this, the stratum lucidum and the stratum corneum, both dead. Layers of cells that are dead. This is where they start to die because of this keratinization process. So every layer below or deeper to the stratum granulosum, the spinosum and the basale, the cells are going to be alive and kicking. Every layer above this, they're going to be dead. This is kind of that transition part. Next, we have the stratum spinosum, and the stratum spinosum is named because under the microscope, with a lot of magnification, these flat pancake cells appear to have these spines attaching to them, and these spines are called desmosomes. And the way I always picture desmosomes is they're kind of like rubber bands. They're just really, really strong rubber bands, real thick rubber bands. So what's the purpose of a rubber band? It's to be strong while under stress or while stretching, right? So if I'm grading your lab quizzes or something and I got 20-something pieces of paper, and then I have another class come in and do the same assignment or quiz or whatever, lab quiz, and another 20 pieces of paper, I need to be able to stretch that rubber band and slide in the other 20 pieces of paper. So now I have 40, right? So that rubber band is able to hold that stretching position while being strong and hopefully not breaking. And that's what these desmosomes do. They allow, there are these junctions between these cells, and they allow stretching and movement to occur without the cells and the tissue becoming damaged. The only other place where we have this type of protein, these desmosomes, are in your heart tissue, your heart cells. And think about what your heart has to do on a 24/7 basis. It fills up with blood, so those cells are stretching, and then they have to forcefully contract and pump that blood out to the rest of your body. So they work as rubber bands. They constantly stretch and contract. And we're also going to have a lot of RNA in this area to help the prep stages of making the keratin. So remember on the previous slide, or the stratum right above this, we have the stratum granulosum. That's where we are going through keratinization. We are pumping the cells full of keratin. Here in the stratum spinosum, we're preparing it. We're kind of getting ready to do that. And remember the very important role of RNA in our cell chapter. RNA is what's going to help us translate that message from a bunch of ACGs and U's, ACGs and U's to the actual amino acid, right? And a bunch of amino acids will make our protein, and that's all keratin is a protein. Then we have the stratum basale. Come, let's get some bacon. So this is the very last stage, or the last part, the deepest part of the epidermis. As soon as you go underneath the stratum basale, you are officially in the dermis. And it is made up of a different type of epithelial tissue, as I pointed out earlier. So 90, 95, 99, whatever percent you want to use, by far the majority of your epidermis is stratified squamous. But this very, very bottom row is made up of simple cuboidal epithelium. And once again, these are stem cells. So they're heavily mitotic. They make your new skin cells. And then what's going to happen is, as these cells get born, get kicked out, they eventually just keep getting pushed by newer cells and newer cells and newer cells, and eventually, like I said, they eventually just flake off in your bed sheets or your pillowcase, whatever. Our next topic is epidermal growth and repair. And remember, the main function of the epidermis and really the skin is protection. So we have this term called turnover time or regeneration time. And what it is, it's basically a measurement of time seeing how long does it take for a cell to be born, a keratinocyte to be born in the stratum basale, and move all the way up the layers, up the stratum spinosum, granulosum, lucidum, corneum, and then eventually flake off. And the average turnover time is about 35 days. So if you're fair-skinned like myself and you go out and get a nice tan during spring break or summer, and then you stop going out in the sun, you'll notice that your tan only lasts for about a month or so, and then it goes away. And the reason why is because you got those nice tan cells at the bottom of your epidermis, and then they flaked off, and your new cells, the melanocytes, don't really change over time. Your genetics is your genetics. So that's kind of how long it takes. If you notice, only about 10% of the cells in the stratum basale actually go through mitosis on a daily basis. So your epidermis grows in clusters. It's not the entire skin making cells every day. It's just portions. So if we have a group of cells, cuboidal stem cells in the stratum basale, they'll go through mitosis on a Tuesday, and then they'll get a week or two off before they go through mitosis again. So it kind of just moves in these different clusters. And you take a look at the last bullet point. If we have a shortened turnover time, we have a callus. So if we're not allowing our skin to have enough time to regenerate, basically we're just putting too much strain or abrasion or pressure on the skin, what will happen is we'll irritate it and we'll get this callus formation, or in bad cases we can actually just break through the skin and get a blister.